on Rabies in 2018 estimated that India had 20,000 human deaths a year from the rabies, accounting for 60% of the rabies death in Asia and 35% globally. Rabies deaths were not evenly distributed throughout the country. One of all rabies deaths were found in Uttar Pradesh, which is 4,300, and nearly three quarter 8,900 were in the seventh central and the southern eastern states: Chhattisgarh, Uttar Pradesh, Odisha, Andhra Pradesh, Bihar, Assam, and Madhya Pradesh. Among larger states, the highest rate of the rabies death were 1 lakh population were in the Chhattisgarh and Uttar Pradesh and Odisha. No rabies death were reported in the study areas from the following states like Kerala, Jammu Kashmir, Jharkhand, Manipur, Meghalaya, and so many. Together, these states represent approximately 7% of the 7% of fourth Indians population. Coming toward the next slide. I think yes, and this. Thank you. The post exposure prophylaxis is the immediate treatment of a bite victim after a rabies exposure. This prevents the virus entry into the central nervous system, which results in imminent death. In the post exposure prophylaxis consists of the extensive washing and local treatment of the bite, wound, or scratch as soon as possible after the suspected exposure, and a course of a potent and effective rabies vaccine that meets WHO standards, and the administration of the rabies immunoglobulin if indicated. Starting the treatment soon after an exposure to the rabies virus can effectively prevent the onset of symptoms and death. Extensive wound washing. The first aid measures include the immediate and through flushing and washing of the wound for the minimum of 15 minutes with soap and water, detergent, povidone iodine, or other substances that remove and kill the rabies virus. India India Metacentric Rabies Survey in 2017, WHO information sheet above, rate of the vaccine reactions, rabies vaccine in June 2012. India J Public Health, and coming toward the next slide. Yes. Firstly, the compliance. Who received the full course of anti-rabies vaccine, that is the scheduled dose of ARVs on the recommended dates. Delay compliance. Who completed the scheduled dose of ARV within 16 days but deviated from the scheduled regimen. And yes, we have the dropout, non-compliance, who had taken at least one dose of ARV after the animal bite but not completed all the scheduled doses. Rabies post-exposure prophylaxis in a routine practice in a view of the new center of the Diseases Control and Prevention and World Health Organization recommendation. A. The retrospective analysis done at the travel clinic. Second, a total of 1,010 patients who received the rabies for 4 dose post-exposure prophylaxis. The shortcoming in the management of these patients were late initiation of rabies prophylaxis in the traveler, administration of human rabies immunoglobulin, and thirdly, antibody level is more than the 0.5 in 6 to 90 patients after the 4 dose of vaccination. Conclusion is the patient do not always receive the optimal rabies post-exposure prophylaxis under the real life conditions. A significant proportion of the patient did not develop adequate antibody level after the 4 dose of vaccination. Coming toward the next slide, back down. New recommendation of the rabies post-exposure prophylaxis We published were the published by the Center of the Disease Control and Prevention and the World Health Organization in 2010. In view of these new recommendations, we investigated the adequacy of the rabies post-exposure prophylaxis among the patient consulting over a travel clinic. Method: a retrospective analysis of the files of all the patients who consulated of rabies PEV at the travel clinic of the University Hospital in Lausanne, Switzerland, between the January 2005 and August 2011 was conducted. Result: a total of 1,010 patients who received rabies VP were indicated. The media age of the patient was 34 years, and 53% were the women. 9 subjects were potentially exposed to the rabies while traveling abroad. Shortcoming in the management of these patients were late initiation of the rabies travel, who waited to seek medical care until returning to the Switzerland administration of the human rabies immunoglobulin to only 7 of 50 travels who sought care abroad and for whom human rabies immunoglobulin was indicated and the antibody level after the 4 doses of the vaccination. Patients do not always receive the optimal rabies prophylaxis under the real life conditions. As I told you, if the patient did not develop the adequate antibody levels after the 4 doses of vaccination, these data indicate that the measurement of the antibody level after 21 days of the ASIN post-exposure prophylaxis regimen is useful in order to verify and adequate the immune response. The study: 4 cases of rabies having the street dogs bite category grade 3. Everyone took vaccination at least 3 doses, but none of them took rabies immunoglobulin. They develop a symptom of rabies with a gap of 15 to 28 days after the bite and admitted to a tertiary care center. One patient was left against some medical advice, and 3 patients were treated according to the modified Milwaukee protocol, but none of them could be saved. So, it may be proposed that the reason of the death may be due to the lack of the administration of rabies immunoglobulin or the failure of the vaccine. Hence, the government may focus on the administration of the complete and quality post-exposure prophylaxis in the case of animal bite. Although the protocol save few lives, it may be further improved or other treatment modalities may be developed for rabies treatment. Coming toward the next slide. Although rabies is 100% fatal disease, it is preventable with appropriate and timely post-exposure prophylaxis. A hospital-based study was conducted among the 360 animal bite patients in the anti-rabies clinic of a Vardhaman Mahavir Medical College and Safdarjung Hospital, New Delhi. Materials and methods: a pre-designed, pre-tested structure cautionary was administered by the interview technique to assess the compliance of the post-exposure prophylaxis among the animal bite patient attending the ARC, which comprised the assessment of wound washing practice adopted by the animal bite patient, administration of the rabies immunoglobulin, and compliance of a full course of anti-rabies vaccine. Result: out of 360 study participant, 131 has washed the wound using the soap and water, and 216 was washed the wound for less than 5 minutes. The compliance of the full course of ARV was seen among 172 study participants, where the 164 were found to be non-compliant and 24 were found to be delayed compliant. Conclusion: it was observed that the considerable segment of the study population did not participate the correct wound washing practice, and only about half of the total study participants were found to be compliant to the full course of ARV. As rabies is a 100% preventable disease, increasing the awareness pertaining to appropriate PEP may prove to be beneficial in improving the compliance of the post-exposure prophylaxis and reducing the burden of mortality. Coming toward the next slide, the complete post-exposure prophylaxis is necessary to prevent the rabies among all the animal bite victims. It is essential for the bite victim to complete the full course of vaccination as recommended for the complete protection. The present study was conducted to determine the compliance rate for the anti-rabies vaccination by both the intramuscular route and the intradermal route to be determined the major constraints. The study may done at two municipal corporations hospital in Bengaluru. India is compliance rate for intramuscular rabies vaccination was 60% and the intradermal rabies vaccination is 77%. The major constraints were the loss of wages, forgotten dates, cost incurred, distance from the hospital. Hence, the present study showed that the compliance to the anti-rabies vaccination for the post-exposure prophylaxis is low. And it is a cause of a concern as the animal bite victim who do not complete the full course of the vaccination are still at the risk of developing rabies. Coming toward the next slide, among 86 non-compliance in intramuscular rabies vaccine group and 54 could be contracted for inquiry regarding their the constraint to complete the course. The major constraint were the loss of wages, forgotten dates, cost incurred, interference with the working hours, school timing, and distance from the hospital. Now, the evolution of the rabies vaccine, the development of the rabies vaccine consent through the four chronological stages. Those are one, neural tissue vaccine, second is the non-neural vaccine, third is the cellular culture based vaccine, and fourth is the recombinant DNA and the protein based vaccine, which is our in 1985 the Pasteur first developed the live attenuated rabies vaccine. However, this vaccine had a various adverse effects such as the neurological disorder and sometimes death. The member of the first generation rabies vaccine includes the vaccine prepared in the neural tissue, includes the Pasteur vaccines in 1885 living and the Huygens vaccine and the Buscaris vaccine and the suckling mouse brain vaccine and the suckling rat brain vaccine and the suckling rabbit brain vaccine. All these vaccines prompt the autoimmune reaction is the reception. Due to these reasons, the second generation non-neural vaccines were developed. These were embryonated egg were the used for the culturing the virus. The fourth generation of the rabies vaccines were developed based on the recommendation DNA technology, where either the DNA vaccine of the protein subunit vaccine were developed. Rabies virus consists of the 5 genes: neuroprotein, phosphoprotein, matrix protein, glycoprotein, and the viral RNA polymerase. And among all these proteins, the G and the N protein are known to be important for the immunogenicity against the rabbit virus. Many approaches are explored since the DNA recombinant technology becomes available to express the immunogenic recombination rabies virus glycoprotein. Unfortunately, in human DNA, vaccines were found to be poorly immunogenic. Coming toward the next slide, novel 3-dose recombinant rabies G protein vaccine. Here we have this. You can see the slide. The first subunit and prepared by VLP platform, first vaccine 3-dose intramuscular regimen, and better compliance due to the easy of the flow regimen and the less versatile. Convenience, ready to use, no reconstitution needed. The rabies is the world's first 3-dose intramuscular rabies vaccine. Rabies Vaccine is a subunit vaccine prepared using the virus-like particles platform where the nanoparticles can be engineered to elect the differentiate the immune response which can increase the vaccine efficacy. Travis is available as ready-to-use vaccine, no reconstitution required before the administration. Currently, 5-dose regimen are recommended for the post-exposure vaccination. Compliance remains a major challenge. With the 5-day schedule in the post-exposure prophylaxis of the rabies, non-compliance rate increase as a dose of duration increase. It is reported as the high as 40% in fifth dose regimen. Rabies vaccine may help to improve the compliance toward the full course of immunization due to the unique advantages like reduced dosage, reducing the dosage schedule 037 days, resulting in less number of the injections and reduce the number of visits of the patient to the hospital. Here you can see the slides, the VLP technology we are showing you. The Here are the slides complete. The recent advantage in the vaccine development have led to the availability of the new year, more cost-effective vaccine that require the fewer dose in the breakthrough achievement. The Cadila Pharmaceutical was developed a novel 3-dose recombinant nanoparticle-based rabies G protein vaccine. The vaccine Travis is prepared by using the virus-like particle technology, the VLP self-assemble for the recombinant G protein, which is produced from the genes clone into the baculovirus, express the vector and expressed in the Spodopetra frugipeda insects cell. The vaccine generates the antibodies against the G protein, which leads to the virus neutralization, as well as prevent the virus attachment to the cell to confer the protection against the rabies. The vaccine has to be administered through the intramuscular route on the day 0, 3, and 7. That's the clinical phase 3 we are showing you. The clinical trial design consists of the 5 study arms. The rabies G protein vaccine arm includes the dosage 10 to 50 microgram and scheduled 037 days. A complete seroprotection was achieved for all the dosage schedule regimen of the day 14 and sustained the same at the day 42. No anaphylactic reactions are reported in this phase 2. Out of total 225 subjects, 40 subjects reported the AIA and headache was the most frequent adverse effect event, followed by the muscle pain and the tendon tiredness. Most frequent body system affected by this was just the general disorder by administration, the site of the condition, musculoskeletal and the connective tissue disorder and the nervous system disorder. On the stage, now I'm showing you the clinical study phase 3 to establish the non-inferiority of a novel 3-dose recombinant G protein vaccine to be administered as a stimulated post-exposure prophylaxis when compared to the 5-dose WHO pre-qualified vaccine for the better safety and immunogenicity. And here is the phase 3 study, multicentric, open label, accessories blind center, specific block, randomized the parallel design. The phase 3 clinical study was conducted among the 800 subjects. The eligible subjects were recommended to the 2:1 for the recombinant G protein vaccine and the reference vaccine. Subject in the recombinant G protein vaccine arm received a 3-dose of vaccine on the day 0, 3, and 7, while the subject in the reference vaccine arms received the 5 dosage of the WHO pre-qualified vaccines on the day 0, 3, 7, 14, and 28. And the results here, firstly, talking about the efficacy as per the WHO recommendation, RVNA titer of the 0.5 international unit per ml in the considered as a seroprotective against the rabies. The seroprotection on the day 14 was found to be the 99.24% and the 97.72% in the recombinant rabies G protein, which is Thrabis. Thrabis vaccine arms are the reference vaccine arm respectively, and the difference are statistically non-significant. Likewise, the day 42, the 98.69% of the subjects in the test vaccines arm Travis and 100% in the reference vaccine were zero, zero positive. The difference was statistically non-significant. Now, talking about the safety, about the 9.9% subject in the recombinant G protein vaccine arm in 17.2% in subject in the reference arm reported the adverse effect. All the adverse effects were mild to moderate in nature, while they resolved without any complication. The most frequent observed local were the pain, redness, swelling at the site of the injection. The systematic were the fever, headache, ear pain, joint pain, and nausea. None of the study subject in either group discontinued the vaccination course due to the no that the serious of the anaphylactic reaction was observed during the entire study. The compliance to complete the course of the vaccine was 99.5% among the 800 participants. The study subjects were healthy up to the day around the ear in both the arms. So here are the conclusions of our study. In conclusion, the novel 3-dose recombinant G protein vaccine was found to be safe and immunogenic and was comparable to the 5-dose of WHO pre-qualified vaccine in the simulated post-exposure prophylaxis. The reduced number of the vaccine dose lead to the reduction in the number of the visit and travel cost as well as increase the compliance, which is important to prevent the rabies and ultimately help in the eliminating the dog mediated human rabies by 2030. Here are the here are the achievements that are gained by the Cadila. The Travis wins the prize over here, the World Awards for the Innovative Pharma of the Year. The Cadila Pharmaceutical, we should congratulate Cadila for achieving this milestone. Cadila Pharmaceutical Rabies Vaccine Travis received the award at a global Golden Globe Tiger Award in Malaysia. I hope you all, if you guys have any query, you can ask.
Dr. Sab, good evening, sir.
Yes, sir.
Dr. Sab, very thank you to your support. And any doctor can ask any question related to the rabies and related to rabies vaccine.
Yes. Here I am like to thank you all the team of the Cadila for giving me this opportunity to explain this a wonderful product to all the doctors, and I hope you all have clear out all your doubts. Here I am have some OPDs so I can clear it out. Thank you everyone.
Thank you, Dr.
Take
Thank you
care.
very much on behalf of Cadila Pharma. I welcome you and I'm very much thankful to you, Doctor. Please.
Okay. Thank you.